Chapter 14 Energy Generation in Mitochondria and Chloroplasts. The fundamental need to generate energy efficiently has had a profound influence on the history of life on Earth. Much of the structure, function, and evolution of cells and organisms can be related to their need for energy. The earliest cells may have produced ATP by breaking down organic molecules left by earlier geochemical processes, using some form of fermentation. Fermentation reactions occur in the cytosol of present-day cells. As discussed in Chapter 13, these reactions use the energy derived from the partial oxidation of energy-rich food molecules to form ATP, the chemical energy currency of cells. But very early in the history of life, a much more efficient method for generating energy and synthesizing ATP appeared. This process is based on the transport of electrons along membranes. Billions of years later, it is so central to the survival of life on Earth that we devote this entire chapter to it. As we shall see, this membrane-based mechanism is used by cells to acquire energy from a wide variety of sources. For example, it is central to the conversion of light energy into chemical bond energy in photosynthesis, and to the aerobic respiration that enables us to use oxygen to produce large amounts of ATP from food molecules. The mechanism we will describe first appeared in bacteria more than 3 billion years ago. The descendants of these pioneering cells crowd every corner and crevice of the land and the oceans with a wild menagerie of living forms, and they survive within eukaryotic cells in the form of chloroplasts and mitochondria. Where we come from and how we are related to other living things are puzzles that have fascinated humans since the beginning of recorded time. The story that we can tell now, worked out through a long chain of scientific investigation, is one of the most dramatic and exciting histories ever told. And we are not yet done. Each year, further discoveries in cell biology enable us to add more details through molecular detective work of dramatically increasing power. Absolutely central to life's progression was the ability to provide an abundant source of energy for cells. In this chapter, we discuss the remarkable mechanism that made this possible. Cells obtain most of their energy by a membrane-based mechanism. The main chemical energy currency in cells is ATP. See Figure 332. In eukaryotic cells, small amounts of ATP are generated during glycolysis in the cytosol, but most ATP is produced by oxidative phosphorylation in mitochondria, as outlined in Chapter 13. The mechanism by which the bulk of ATP is generated in the mitochondria differs from the way in which ATP is produced by glycolysis in that it involves a membrane. Oxidative phosphorylation depends on electron transport within the mitochondrial membrane and the transport of ions across it. The same type of ATP generating process occurs in the plasma membrane of bacteria. The membrane-based mechanism for making ATP arose very early in life's history and was so successful that its essential features have been retained in the long evolutionary journey from early prokaryotes to modern cells. In photosynthetic bacteria, plants, and algae, a related membrane-based process produces ATP during photosynthesis. The membrane-based process for making ATP consists of two linked stages. Both are carried out by protein complexes in the membrane. Stage 1. Electrons derived from the oxidation of food molecules, as discussed in Chapter 13, or from other sources discussed later, are transferred along a series of electron carriers, called an electron transport chain, embedded in the membrane. These electron transfers release energy that is used to pump protons, H+, derived from the water that is ubiquitous in cells, across the membrane and thus generate an electrochemical proton gradient. Figure 14.1a. An ion gradient across a membrane is a form of stored energy that can be harnessed to do useful work when the ions are allowed to flow back across the membrane down their gradient, as discussed in Chapter 12. Stage 2. H+ flows back down its electrochemical gradient through a protein complex called ATP synthase, which catalyzes the energy-requiring synthesis of ATP from ADP and inorganic phosphate, P subscript I. This ubiquitous enzyme serves the role of a turbine, permitting the proton gradient to drive the production of ATP. Figure 14.1b. The linkage of electron transport, proton pumping, and ATP synthesis was called the Chemiosmotic Hypothesis when it was first proposed in the 1960s, because of the link between the chemical bond-forming reactions that synthesize ATP, chemi, and the membrane transport processes, osmotic, from the Greek osmos, to push. It is now known as chemiosmotic coupling. Chemiosmotic mechanisms allow cells to harness the energy of electron transfers in much the same way that the energy stored in a battery can be harnessed to do useful work. Figure 14.2. Chemiosmotic coupling first evolved in bacteria. Aerobic eukaryotic cells appear to have adopted the bacterial chemiosmotic mechanisms intact, first by engulfing aerobic bacteria to form mitochondria, and somewhat later, in the lineages leading to algae and plants, by engulfing cyanobacteria to form chloroplasts, as described in Chapter 1. See figures 119 and 121. In this chapter, we shall consider energy generation in both mitochondria and chloroplasts, emphasizing the common principles by which proton gradients are created and used in these organelles and in the plasma membranes of bacteria. We start by describing the structure and function of mitochondria, looking in detail at the events that occur in the mitochondrial membrane to create the proton gradient and generate ATP. We next consider photosynthesis in the chloroplasts of plant cells. Finally, we trace the evolutionary pathways that gave rise to these mechanisms of energy generation. By examining the lifestyles of a variety of single-celled organisms, including those that might resemble our early ancestors, we can begin to see the role that chemiosmotic coupling has played in the rise of complex eukaryotes and in the development of all life on Earth. Mitochondria and oxidative phosphorylation. Mitochondria are present in nearly all eukaryotic cells, in plants, animals, and most eukaryotic microorganisms, and most of a cell's ATP is produced in these organelles. Without them, present-day eukaryotes would be dependent on the relatively inefficient process of glycolysis for all of their ATP production, and it seems unlikely that complex multicellular organisms would have been supported in this way. When glucose is converted to pyruvate by glycolysis, only 2 molecules of ATP are produced per glucose molecule, less than 10% of the total free energy potentially available. By contrast, in mitochondria, the metabolism of sugars is completed and the energy released is harnessed so efficiently that about 30 molecules of ATP are produced for each molecule of glucose oxidized. Defects in mitochondrial function can have serious repercussions for any organism. Consider, for example, an inherited disorder called myoclonic epilepsy and ragged red fiber disease, MRF. This disease, caused by a mutation in one of the mitochondrial transfer RNA, tRNA genes, is characterized by a decrease in synthesis of the mitochondrial proteins required for electron transport and ATP production. As a result, patients with this disorder typically experience muscle weakness or heart problems, from effects on cardiac muscle, and epilepsy or dementia, from effects on nerve cells. Muscle and nerve cells suffer most when mitochondria are defective, because they need especially large amounts of ATP to function well. The same metabolic reactions that occur in mitochondria also take place in aerobic bacteria, which do not possess these organelles. In these organisms, the plasma membrane carries out the chemiosmotic coupling. Of course, a bacterial cell also has to carry out many other functions. The mitochondrion, by contrast, has become highly specialized for energy generation. A mitochondrion contains an outer membrane, an inner membrane, and two internal compartments. Mitochondria are generally similar in size and shape to bacteria, although these attributes can vary depending on the cell type. They contain their own DNA and RNA, and a complete transcription and translation system, including ribosomes, which allows them to synthesize some of their own proteins. Time-lapse movies of living cells reveal mitochondria as remarkably mobile organelles, constantly changing shape and position. Present in large numbers, 1,000 to 2,000 in a liver cell, for example, these organelles can form long, moving chains in association with the microtubules of the cytoskeleton, discussed in Chapter 17. In other cells, they remain fixed in one location to target ATP directly to a site of unusually high ATP consumption. In a heart muscle cell, for example, mitochondria are located close to the contractile apparatus, whereas in a sperm, they are wrapped tightly around the motile flagellum. Figure 14.3. The number of mitochondria present in different cell types varies dramatically and can change with the energy needs of the cell. In skeletal muscle cells, for example, the number of mitochondria may increase 5 to 10-fold due to mitochondrial growth and division that occur if the muscle cell has been repeatedly stimulated to contract. Any individual mitochondrion is bounded by two highly specialized membranes, one surrounding the other, that play a crucial part in its activities. The outer and inner mitochondrial membranes create two mitochondrial compartments, a large internal space called the matrix and the much narrower intermembrane space, figure 14.4. If purified mitochondria are gently processed and fractionated into separate components by differential centrifugation, see panel 4.4, pages 164 to 165, the biochemical composition of each of the two membranes and of the spaces enclosed by them can be determined. Each contains a unique collection of proteins. The outer membrane contains many molecules of a transport protein called porin, which, as described in Chapter 11, forms wide aqueous channels through the lipid bilayer. As a result, the outer membrane is like a sieve that is permeable to all molecules of 5,000 Daltons or less, including small proteins. This makes the intermembrane space chemically equivalent to the cytosol with respect to the small molecules it contains. In contrast, the inner membrane, like other membranes in the cell, is impermeable to the passage of ions and most small molecules, except where a path is provided by membrane transport proteins. The mitochondrial matrix therefore contains only molecules that can be selectively transported into the matrix across the inner membrane, and its contents are highly specialized. The inner mitochondrial membrane is the site of electron transport and proton pumping, and it contains the ATP synthase. Most of the proteins embedded in the inner mitochondrial membrane are components of the electron transport chains required for oxidative phosphorylation. This membrane also contains a variety of transport proteins that allow the entry of selected small molecules, such as pyruvate and fatty acids, into the matrix. The inner membrane is usually highly convoluted, forming a series of infoldings, known as cristae, that project into the matrix space to greatly increase the surface area of the inner membrane. See Figure 14.4. These folds provide a large surface on which ATP synthesis can take place. In a liver cell, for example, the inner mitochondrial membranes of all the mitochondria constitute about 1/3 of the total membranes of the cell, and the number of cristae is 3 times greater in a mitochondrion of a cardiac muscle cell than in a mitochondrion of a liver cell. The citric acid cycle generates high-energy electrons. Mitochondria use both pyruvate and fatty acids as fuel, the pyruvate coming mainly from glucose and other sugars, and the fatty acids from fats. These fuel molecules are transported across the inner mitochondrial membrane and then converted to the crucial metabolic intermediate acetyl-CoA by enzymes located in the mitochondrial matrix. See Figure 13.10. The acetyl groups in acetyl-CoA are then oxidized in the matrix via the citric acid cycle. See Panel 13.2, pages 442 to 443. The cycle converts the carbon atoms in acetyl-CoA to CO2, which is released from the cells as a waste product. In addition, the cycle generates high-energy electrons, carried by the activated carrier molecules, NADH and FADH2. Figure 14.5. Although the citric acid cycle is considered to be part of aerobic metabolism, it does not itself use molecular oxygen, O2. Oxygen is directly consumed only in the final catabolic reactions that take place on the inner mitochondrial membrane, as we see next. A chemiosmotic process converts the energy from activated carrier molecules into ATP. Nearly all the energy available from burning carbohydrates, fats, and other foodstuffs in the early stages of their oxidation is initially saved in the form of the activated carrier molecules generated during glycolysis and the citric acid cycle, NADH and FADH2. These carrier molecules donate their high-energy electrons to the electron transport chain in the mitochondrial membrane, and thus become oxidized to NAD+ and FAD. The electrons are quickly passed along the chain to molecular oxygen O2 to form water, H2O. The passage of the high-energy electrons along the electron transport chain releases energy that is harnessed to pump protons across the inner mitochondrial membrane. Figure 14.6. The resulting proton gradient, in turn, drives the synthesis of ATP. The full sequence of reactions is shown in Figure 14.7. The inner mitochondrial membrane, thus serves as a device that converts the energy contained in the high-energy electrons of NADH into the high-energy phosphate bond of ATP. Figure 14.8. This chemiosmotic mechanism of ATP synthesis is called oxidative phosphorylation because it involves both the consumption of O2 and the addition of a phosphate group to ADP to form ATP. Although chemiosmotic coupling escaped detection for many years, the vast majority of living organisms use this mechanism to generate ATP. The source of the electrons that power the proton pumping differs widely between different organisms and different processes. In aerobic respiration in mitochondria and aerobic bacteria, the electrons are ultimately derived from glucose or fatty acids. In photosynthesis, the required electrons are derived from the action of light on the green pigment chlorophyll, and many bacteria use inorganic substances such as hydrogen, iron, and sulfur as the source of the high-energy electrons that they need to make ATP. The electron transport chain pumps protons across the inner mitochondrial membrane. The electron transport chain, or respiratory chain, that carries out oxidative phosphorylation is present in many copies in the inner mitochondrial membrane. Each chain contains over 40 proteins, most of which are embedded in the lipid bilayer and function only in the intact membrane, making them difficult to study. However, the components of the electron transport chain, like other membrane proteins, can be solubilized using non-ionic detergents. See Figure 11.27, purified and then reconstituted in operational form in small membrane vesicles. Such studies reveal that most of the proteins involved in the mitochondrial electron transport chain are grouped into 3 large respiratory enzyme complexes, each containing multiple individual proteins. Each complex includes transmembrane proteins that hold the entire complex firmly in the inner mitochondrial membrane. The 3 respiratory enzyme complexes, in the order in which they receive electrons, are 1. The NADH dehydrogenase complex, 2. The cytochrome BC subscript 1 complex, and 3. The cytochrome oxidase complex. Each contains metal ions and other chemical groups that form a pathway for the passage of electrons through the complex. The respiratory complexes are the sites of proton pumping, and each can be thought of as a protein machine that pumps protons across the membrane as electrons are transferred through it. Electron transport begins when a hydride ion, H-, is removed from the NADH and is converted into a proton and 2 high-energy electrons, H- to H++ plus 2E-, as shown previously in Figure 14.5. This reaction is catalyzed by the first of the respiratory enzyme complexes, the NADH dehydrogenase, which accepts electrons from NADH, Figure 14.9. The electrons are then passed along the chain to each of the other enzyme complexes in turn, using mobile electron carriers to ferry electrons between complexes. The transfer of electrons along the chain is energetically favorable. The electrons start out at very high energy and lose energy at each transfer step, eventually entering Cytochrome Oxidase, where they combine with a molecule of O2 to form water. This is the oxygen-requiring step of cellular respiration, and it consumes nearly all of the oxygen that we breathe. Proton pumping creates a steep electrochemical proton gradient across the inner mitochondrial membrane. Without a mechanism for harnessing the energy released by electron transfers, this energy would simply be liberated as heat. But cells utilize much of the energy of electron transfer by having the transfers take place within proteins that are capable of pumping protons. In this way, the energetically favorable flow of electrons along the electron transport chain results in the pumping of protons across the membrane out of the mitochondrial matrix and into the space between the inner and outer mitochondrial membranes. See Figure 14.9. Later in the chapter, we review the detailed molecular mechanisms that couple electron transport to the movement of protons. For now, we focus on the consequences of this nifty biological maneuver. First, the active pumping of protons generates a gradient of H+ concentration, a pH gradient, across the inner mitochondrial membrane, where the pH is about 0.5 unit higher in the matrix, around pH 7.5 than in the intermembrane space, which is close to 7, the same pH as the cytosol. Second, proton pumping generates a membrane potential across the inner mitochondrial membrane, with the inside, the matrix side, negative and the outside positive as a result of the net outflow of H+. As discussed in Chapter 12, the force driving the passive flow of an ion across a membrane is proportional to the electrochemical gradient for the ion across the membrane. This, in turn, depends on the voltage across the membrane, which is measured as the membrane potential, and on the concentration gradient of the ion. See Figure 12.7. Because protons are positively charged, they will move more readily across a membrane if the membrane has an excess of negative electrical charges on the other side. In the case of the inner mitochondrial membrane, the pH gradient and membrane potential work together to create a steep electrochemical proton gradient that makes it energetically very favorable for H+ to flow back into the mitochondrial matrix. In the energy-producing membranes we discuss in this chapter, the membrane potential adds to the driving force pulling H+ back across the membrane, which is called the proton motive force. Hence, the membrane potential increases the amount of energy stored in the proton gradient. Figure 14.10. The electrochemical proton gradient drives ATP synthesis. As explained previously, the electrochemical proton gradient across the inner mitochondrial membrane is used to drive ATP synthesis. The device that makes this possible is a large enzyme called ATP Synthase, which is also embedded in the inner mitochondrial membrane. ATP synthase creates a hydrophilic pathway across the inner mitochondrial membrane that allows protons to flow back across the membrane down their electrochemical gradient. Figure 14.11. As these ions thread their way through the enzyme, they are used to drive the energetically unfavorable reaction between ADP and P subscript I that makes ATP. See Figure 224. The ATP synthase is of ancient origin. The same enzyme occurs in the mitochondria of animal cells, the chloroplasts of plants and algae, and in the plasma membrane of bacteria. ATP synthase is a large multi-subunit protein, Figure 14.12. A large enzymatic portion, shaped like a lollipop head, projects into the matrix and carries out the phosphorylation reaction. This enzymatic structure is attached through a thinner multi-subunit stalk to a transmembrane proton carrier. As protons pass through a narrow channel within the carrier, Their movement causes the stalk to spin rapidly within the head, inducing the head to make ATP. See Figure 1412a. The synthase essentially acts as an energy-generating molecular motor, converting the energy of proton flow down a gradient into the mechanical energy of two sets of proteins rubbing against one another, rotating stalk proteins pushing against stationary head proteins. The movement of the stalk changes the conformation of subunits within the head. This mechanical deformation gets converted into chemical bond energy as the subunits produce ATP. This marvelous device can produce more than 100 molecules of ATP per second, and about 3 protons need to pass through the synthase to make each molecule of ATP. The ATP synthase is a reversible coupling device. It can either harness the flow of protons down their electrochemical gradient to make ATP, its normal role in mitochondria and the plasma membrane of bacteria growing aerobically, or use the energy of ATP hydrolysis to pump protons across a membrane. Figure 1413. In the latter mode, ATP synthase functions like the H+ pumps described in Chapter 12. Whether the ATP synthase primarily makes or consumes ATP depends on the magnitude of the electrochemical proton gradient across the membrane in which it sits. In many bacteria that can grow either aerobically or anaerobically, the direction in which the ATP synthase works is routinely reversed when the bacterium runs out of O2. At this point, the ATP synthase uses some of the ATP generated inside the cell by glycolysis to pump protons out of the cell, creating the proton gradient that the bacterial cell needs to import its essential nutrients by coupled transport, as we see next. Coupled transport across the inner mitochondrial membrane is also driven by the electrochemical proton gradient. The synthesis of ATP is not the only process driven by the electrochemical proton gradient. In mitochondria, many charged molecules such as pyruvate, ADP, and P⁻I are pumped into the matrix from the cytosol, while others, such as ATP, must be moved in the opposite direction. Carrier proteins that bind these molecules can couple their transport to the energetically favorable flow of H+ into the mitochondrial matrix. Pyruvate and inorganic phosphate, P⁻I, for example, are individually co-transported inward with H+ as the latter moves down its electrochemical gradient into the matrix. Other transporters take advantage of the fact that the electrochemical proton gradient generates a membrane potential, such that the matrix side of the inner mitochondrial membrane is more negatively charged than the intermembrane space on the other side. An antiport carrier protein exploits this voltage gradient to expel ATP from and import ADP to the mitochondrial matrix. Because an ATP molecule has one more negative charge than ADP, swapping these nucleotides results in the movement of one negative charge out of the mitochondrion. This nucleotide exchange, which sends ATP to the cytosol, is thus driven by the charge difference across the inner mitochondrial membrane. Figure 1414. In eukaryotic cells, therefore, the electrochemical proton gradient is used to drive both the formation of ATP and the transport of certain metabolites across the inner mitochondrial membrane. In bacteria, the proton gradient across the bacterial plasma membrane serves all of these functions. But in bacteria, this gradient is also an important source of directly usable energy. In motile bacteria, a flow of protons into the cell drives the rapid rotation of the bacterial flagellum, which propels the bacterium along. Movie 14.5. Oxidative phosphorylation produces most of the cell's ATP. As we mentioned earlier, glycolysis on its own produces a net yield of 2 molecules of ATP for every molecule of glucose, whereas the complete oxidation of glucose, which includes glycolysis and oxidative phosphorylation, generates about 30 ATPs. In glycolysis, it is obvious where those ATP molecules came from. 2 molecules of ATP are consumed early in the process, and 4 molecules of ATP are produced toward the end. See Figure 13.3. But for oxidative phosphorylation, the accounting is less straightforward, because the ATPs are not produced directly, as they are in glycolysis. Instead, they are produced from the energy carried by NADH and FADH2, which are generated during glycolysis and the citric acid cycle. These activated carrier molecules donate their electrons to the electron transport chain that lies in the inner mitochondrial membrane. These movement of these electrons along the respiratory chain fuels the formation of the proton gradient, which in turn powers the production of ATP. How much ATP each carrier molecule ultimately produces depends on several factors, including where its electrons enter the respiratory chain. The NADH molecules produced during the citric acid cycle, which takes place inside the mitochondria, pass their electrons to Nadh Dehydrogenase, the first respiratory enzyme complex in the chain. These electrons then pass from one enzyme complex to the next, promoting the pumping of protons across the inner mitochondrial membrane at each step along the way. These NADH molecules provide energy for the net formation of about 2.5 molecules of ATP. See question 14.5 and its answer. The FADH2 generated during the citric acid cycle, on the other hand, produces a net of only 1.5 molecules of ATP. This is because FADH2 molecules bypass the NADH dehydrogenase complex and pass their electrons to the membrane-embedded mobile-carried ubiquinone. See Figure 14.9. These electrons enter further down the respiratory chain, and they therefore promote the pumping of fewer protons and generate less ATP. Table 14.1 provides a complete accounting of the ATP produced by the oxidation of each molecule of glucose. The oxidation of fatty acids also produces large amounts of NADH and FADH2, which in turn produce large amounts of ATP via oxidative phosphorylation. See Figures 13.9 and 13.10. Thus, the vast majority of the ATP produced in an animal cell is produced by chemiosmotic mechanisms on the mitochondrial membrane. The rapid conversion of ADP to ATP in mitochondria maintains a high ATP-ADP ratio in cells. As a result of the co-transport process discussed earlier, ADP molecules produced by ATP hydrolysis in the cytosol are rapidly drawn back into mitochondria for recharging, and the bulk of the ATP molecules formed in the mitochondrial matrix by oxidative phosphorylation are pumped into the cytosol where they are needed. A small amount of ATP is used within the mitochondrion itself to power the replication of its DNA, protein synthesis, and other energy-consuming reactions. All in all, a typical ATP molecule in the human body shuttles out of the mitochondrion and back into it as ADP for recharging more than once a minute. keeping the concentration of ATP in the cell about 10 times higher than that of ADP. As discussed in Chapter 3, biosynthetic enzymes often drive energetically unfavorable reactions by coupling them to the energetically favorable hydrolysis of ATP. See Figure 333a. The ATP pool is thus used to drive cellular processes in much the same way that a battery can drive an electric engine. If the activity of the mitochondria were halted, ATP levels would fall and the cell's battery would run down. Eventually, energetically unfavorable reactions could no longer take place and the cell would die. The poison cyanide, which blocks electron transport in the inner mitochondrial membrane, causes death in exactly this way. Molecular mechanisms of electron transport and proton pumping. We have already considered in general terms how a mitochondrion couples electron transport to the generation of ATP. We now examine in more detail the molecular mechanisms that underlie its membrane-based energy conversion processes. In doing so, we will also be accomplishing a larger purpose. As emphasized at the beginning of this chapter, very similar energy conversion devices are used by mitochondria, chloroplasts, and bacteria, and the basic principles that we shall discuss next therefore underlie the function of nearly all living things. For many years, the reason that electron transport chains were embedded in membranes eluded the biochemists who were struggling to understand them. The process of chemiosmotic coupling entails an interplay between chemical and electrical forces that is not easy to decipher at a molecular level. The puzzle was essentially solved as soon as the fundamental role of transmembrane proton gradients in energy generation was proposed in the early 1960s. However, the idea was so novel that it was not widely accepted until many years later, after additional supporting evidence had accumulated from experiments designed to test rigorously the chemiosmotic hypothesis. See How We Know, pages 468 to 469. Although investigators today are still unraveling the details of chemiosmotic coupling at the atomic level, the fundamentals are now clear. In this part of the chapter, we shall look at some of the principles that underlie the electron transport process and explain in detail how it can generate a proton gradient. Protons are readily moved by the transfer of electrons. Although protons resemble other positive ions, such as Na+ and K+ in the way they move across membranes, in some respects they are unique. Hydrogen atoms are by far the most abundant type of atom in living organisms, and are plentiful not only in all carbon-containing biological molecules, but also in the water molecules that surround them. The protons in water are highly mobile, flickering through the hydrogen-bonded network of water molecules by rapidly dissociating from one water molecule in order to associate with its neighbor. Thus, water, which is everywhere in cells, serves as a ready reservoir for donating and accepting protons. Whenever a molecule is reduced by acquiring an electron, the electron, E-, brings with it a negative charge. In many cases, this charge is rapidly neutralized by the addition of a proton from water, so that the net effect of the reduction is to transfer an entire hydrogen atom, H+ plus E-, figure 15. Similarly, when a molecule is oxidized, the hydrogen atom can be readily dissociated into its constituent electron and proton, allowing the electron to be transferred separately to a molecule that accepts electrons, while the proton is passed to the water. Therefore, in the membrane in which electrons are being passed along an electron transport chain, it is a relatively simple matter, in principle, to pump protons from one side of the membrane to another. All that is required is that the electron carrier be arranged in the membrane in a way that causes it to pick up a proton from one side of the membrane when it accepts an electron, while releasing the proton on the other side of the membrane as the electron is passed on to the next carrier molecule in the chain. Figure 1416. The redox potential is a measure of electron affinities. The proteins of the respiratory chain guide the electrons so that they move sequentially from one enzyme complex to another, with no short circuits that skip a complex. Each electron transfer is an oxidation-reduction reaction, as described in Chapter 3. The molecule or atom donating the electron becomes oxidized, while the receiving molecule or atom becomes reduced. See pages 87 to 88. Electrons will pass spontaneously from molecules that have a relatively low affinity for their available electrons and thus lose them easily, to molecules with a higher electron affinity. For example, NADH with its high energy electrons has a low electron affinity, so that its electrons are readily passed to the Nadh dehydrogenase. The electrical batteries of our common experience are based on similar electron transfers between two chemical substances with different electron affinities. In biochemical reactions, any electrons removed from one molecule are always passed to another, so that whenever one molecule is oxidized, another is reduced. Like any other chemical reactions, the tendency of such oxidation-reduction reactions or redox reactions to proceed spontaneously depends on the free energy change, delta G, for the electron transfer, which in turn depends on the relative affinities of the two molecules for electrons. The role of free energy in chemical reactions is discussed in Chapter 3, pages 91 to 92. Because electron transfers provide most of the energy for living things, it is worth spending a little time to understand them. Many readers are already familiar with acids and bases, which donate and accept protons. See panel 2.2, pages 66 to 67. Acids and bases exist in conjugate acid-base pairs, where the acid is readily converted into the base by the loss of a proton. For example, acetic acid, CH3COOH, is converted into its conjugate base, CH3COO-, in the reaction CH3COOH to CH3COO- plus H+. In exactly the same way, pairs of compounds such as NADH and NAD+ are called redox pairs, because NADH is converted to NAD+ by the loss of electrons in the reaction, NADH to NAD+ plus H+ plus 2E-. NADH is a strong electron donor because its electrons are held in a high energy linkage. The delta G for passing its electrons to many other molecules is favorable. Conversely, it is difficult to form the high energy linkage in NADH, so its partner, NAD+, is of necessity a weak electron acceptor. The tendency to transfer electrons from any redox pair can be measured experimentally. All that is required is to form an electrical circuit that links a 1-to-1 equimolar mixture of the redox pair to a second redox pair that has been arbitrarily selected as a reference standard, so that the voltage difference between them can be measured. Panel 14.1, page 471. This voltage difference is defined as the redox potential. As defined, electrons will move spontaneously from a redox pair such as NADH, NAD+, with a low redox potential, a low affinity for electrons, to a redox pair such as O2H2O with a high redox potential, a high affinity for electrons. Thus, NADH is a good molecule to donate electrons to the respiratory chain, while O2 is well suited to act as the sink for electrons at the end of the pathway. As explained in panel 14.1, the difference in redox potential, delta E subscript 0 prime, is a direct measure of the standard free energy change, delta G degrees, for the transfer of an electron from one molecule to another. In fact, delta E subscript 0 prime is simply equal to delta G degrees times a negative number that is a constant. Electron transfers release large amounts of energy. As just discussed, those pairs of compounds that have the most negative redox potential, E subscript 0 prime, have the weakest affinity for electrons and therefore the strongest tendency to donate electrons. Conversely, and therefore, the strongest tendency to donate electrons. Conversely, those pairs that have the most positive redox potentials have the strongest affinity for electrons and therefore the strongest tendency to accept electrons. A 1-to-1 mixture of NADH and NAD+ has a redox potential of negative 3020 millivolts, indicating that NADH has a strong tendency to donate electrons. A 1-to-1 mixture of H2O and 1/2 O2 has a redox potential of plus 820 millivolts, indicating that O2 has a strong tendency to accept electrons. The difference in redox potential between these two pairs is 1.14 volts, 1140 millivolts, which means that the transfer of each electron from NADH to O2 under these standard conditions is enormously favorable. The delta G degrees equals negative 26.2 kilocals per mole per reaction, or negative 52.4 kilocals per mole for the 2 electrons transferred per NADH molecule. See panel 14.1. If we compare this free energy change with that for the formation of the phosphoanhydride bonds in ATP, delta G degrees equals plus 7.3 kilocals per mole. See figure 13.7. We see that more than enough energy is released by the oxidation of one NADH molecule to synthesize several molecules of ATP from ADP and P subscript I. Living systems could certainly have evolved enzymes that would allow NADH to donate electrons directly to O2 to make water in the reaction. 2H plus 2E minus plus 1/2 O2 to H2O. But because of the huge free energy drop, this reaction would proceed with almost explosive force and nearly all of the energy would be released as heat. Instead, the energetically favorable reaction 2H plus 2E minus plus 1/2 O2 to H2O is made to occur in many small steps, enabling nearly half of the released energy to be stored rather than being lost to the environment as heat. Metals tightly bound to proteins form versatile electron carriers. Within each of the 3 respiratory enzyme complexes, electrons move mainly between metal atoms that are tightly bound to the proteins, traveling by skipping from one metal ion to another one with a greater affinity for electrons. In contrast, electrons are carried between the different respiratory complexes by molecules that diffuse along the lipid bilayer, picking up electrons from one complex and delivering them to another in an orderly sequence. In both the respiratory and photosynthetic electron transport chain, one of these carriers is a quinone, a small hydrophobic molecule that dissolves in the lipid bilayer. In the mitochondrial respiratory chain, the quinone is called ubiquinone. Quinones are the only electron carriers in electron transport chains that can function without being tightly bound to a protein. Ubiquinone picks up electrons from the Nadh dehydrogenase complex and delivers them to the cytochrome BC1 complex. See figure 14.9. Ubiquinone can pick up or donate either 1 or 2 electrons, and it picks up 1 H plus from the surroundings with each electron that it carries. Figure 14.19. Its redox potential of plus 30 millivolts places ubiquinone about 1/4 of the way down the chain from NADH in terms of energy loss. Figure 14.20. Ubiquinone can also receive electrons directly from the FADH2 generated by the citric acid cycle or by fatty acid oxidation. Because these electrons bypass NADH hydrogenase, which is one of the proton pumps in the electron transport chain, they cause less proton pumping than do the 2 electrons transported from NADH. The rest of the electron carriers in the electron transport chain are either small molecules or metal-containing groups that are all tightly bound to proteins. To get from NADH to ubiquinone, for example, the electrons are passed inside the NADH dehydrogenase complex between a flavin group, see figure 13.12 for its structure, bound to one of the proteins and a set of iron-sulfur centers of increasing redox potentials. The final iron-sulfur center in the dehydrogenase donates its electrons to ubiquinone. Iron-sulfur centers have relatively low affinities for electrons and thus are prominent in the early part of the electron transport chain. Later in the pathway from ubiquinone to O2, iron atoms in heme groups that are tightly bound to cytochrome proteins are commonly used as electron carriers, as in the cytochrome BC1 and cytochrome oxidase complexes. The cytochromes constitute a family of colored proteins, hence their name from the Greek chroma, color. Each contains one or more heme groups whose iron atom changes from the ferric Fe3+ to the ferrous Fe2+ state whenever it accepts an electron. As one would expect, the various cytochromes increase in redox potential as one progresses down the mitochondrial electron transport chain towards O2. The structure of cytochrome C, a small protein that shuttles electrons between the cytochrome BC1 complex and the cytochrome oxidase complex, is shown in figure 14.21. Its redox potential is plus 230 millivolts. At the very end of the respiratory chain, just before O2, the electron carriers are those in the cytochrome oxidase complex. The carriers here are either iron atoms in heme groups or copper atoms that are tightly bound to the complex in specific ways that give them a high redox potential. Cytochrome oxidase catalyzes the reduction of molecular oxygen. Cytochrome oxidase is a protein complex that received electrons from cytochrome C, thus oxidizing it, hence the name cytochrome oxidase. It then donates these electrons to O2. In brief, 4 electrons from cytochrome C and 4 protons from the aqueous environment are added to each O2 molecule in the reaction 4E- plus 4H plus O2 to 2H2O. In addition to the protons that couple with O2, 4 other protons are pumped across the membrane during electron transfer, further increasing the electrochemical proton gradient. Of course, for proton pumping to occur, it must be coupled in some way to energetically favorable reactions. In the case of cytochrome oxidase, The energy comes from the transfer of a series of 4 electrons to an O2 molecule that is bound tightly to the protein. These electron transfers drive allosteric changes in the conformation of the protein that move protons out of the mitochondrial matrix. At its active site, where O2 is bound, cytochrome oxidase contains a complex of a heme iron atom juxtaposed with a tightly bound copper atom. Figure 1422. It is here that nearly all of the oxygen we breathe is used, serving as a final repository for the electrons that NADH donated at the start of the electron transport chain. Oxygen is useful as an electron sink because of its very high affinity for electrons. However, once O2 picks up one electron, it forms the superoxide radical O2-. This radical is dangerously reactive and will avidly take up another 3 electrons wherever it can find them, a tendency that could cause serious damage to nearby DNA, proteins, and lipid membranes. One of the roles of cytochrome oxidase is to hold on tightly to an oxygen molecule until all 4 electrons needed to convert it to 2 H2O molecules are in hand, thereby preventing a random attack on cellular macromolecules by superoxide radicals, damage that has been postulated to be a cause of human aging. The evolution of cytochrome oxidase was crucial to the formation of cells that could use O2 as an electron acceptor, and today this protein is estimated to account for 90% of the total uptake of O2 in humans. This protein complex is therefore essential for all aerobic life. The poisons cyanide and azide are extremely toxic because they bind tightly to the cell's cytochrome oxidase complexes to stop electron transport, thereby greatly reducing ATP production. The mechanism of H+ pumping can be studied in atomic detail. The detailed mechanisms by which electron transport is coupled to H+ pumping is different in each of the 3 different respiratory enzyme complexes. For example, ubiquinone has a central role in proton pumping through the cytochrome BC1 complex, as the quinone picks up an H+ from the aqueous medium along with each electron it carries and liberates it when it releases the electron. See figure 1419. Because ubiquinone is freely mobile in the lipid bilayer, it can accept electrons near the inside surface of the membrane and donate them to the cytochrome BC1 complex near the outside surface. Thus, ubiquinone transfers 1 H+ across the bilayer for every electron it transports. However, 2 protons are pumped per electron in the cytochrome BC1 complex, and there is good evidence for a so-called Q cycle, in which ubiquinone is recycled through the protein complex in a complicated but ordered way that makes this 2-for-1 transfer possible. Allosteric changes in protein conformations can also pump H+. For both the Nadh dehydrogenase complex and the cytochrome oxidase complex, electron transport drives sequential allosteric changes in the protein that cause it to pump H+ across the mitochondrial inner membrane. A general mechanism for this type of H+ pumping is presented in figure 1423. Respiration is amazingly efficient. The free energy changes for burning fats and carbohydrates directly to CO2 and H2O can be compared with the total amount of energy generated and stored in the phosphate bonds of ATP during the corresponding biological oxidations. When this is done, one finds that the efficiency with which oxidation energy is converted into ATP bond energy is often greater than 40%. This is considerably better than the efficiency of most non-biological energy conversion devices. If cells worked only with the efficiency of an electric motor or a gasoline engine, 10 to 20%, an organism would have to eat voraciously to maintain itself. Moreover, because wasted energy is liberated as heat, large organisms, including ourselves, would need more efficient mechanisms than they presently have for giving up heat to the environment. Students sometimes wonder why the chemical interconversions in cells follow such complex pathways. The oxidation of sugars to CO2 plus H2O could certainly be accomplished more directly, eliminating the citric acid cycle and many of the steps in the respiratory chain. This would make respiration easier for students to learn, but it would be a disaster for the cell. Oxidation produces huge amounts of free energy, which can be utilized efficiently only in small bits. Biological oxidative pathways involve many intermediates, each differing only slightly from its predecessor. The energy released is thereby parceled out into small packets that can be efficiently converted to high-energy bonds in useful molecules, such as ATP and NADH, by means of coupled reactions. See Figure 13.1. Having seen how chemiosmotic coupling is used to generate ATP in mitochondria, we now look at how it harnesses light energy for the generation of ATP in chloroplasts. Chloroplasts and photosynthesis. Virtually all of the organic material required by present-day living cells is produced by photosynthesis, the series of light-driven reactions that creates organic molecules from atmospheric carbon dioxide, CO2. Plants, algae, and the most advanced photosynthetic bacteria, such as the cyanobacteria, use electrons from water and the energy of sunlight to convert atmospheric CO2 into organic compounds. In the course of splitting water, they liberate into the atmosphere vast quantities of O2 gas. This oxygen is in turn required for cellular respiration, not only in animals, but also in plants and many bacteria. Thus, the activity of early photosynthetic bacteria, which fill the atmosphere with oxygen, enabled the evolution of life forms that use aerobic metabolism to make their ATP. Figure 14.24. In plants, photosynthesis is carried out in a specialized intracellular organelle, the chloroplast, which contains light-capturing pigments such as the green pigment chlorophyll. All green parts of a plant contain chloroplasts, but for most plants, the leaves are the major sites of photosynthesis. Chloroplasts perform photosynthesis during the daylight hours. The process produces ATP and NADPH, which in turn are used to convert CO2 into sugar inside the chloroplast. Thus, we begin our discussion of photosynthesis by describing the structure of this specialized organelle. Chloroplasts resemble mitochondria but have an extra compartment. Chloroplasts carry out their energy interconversions by means of proton gradients in much the same way that mitochondria do.